Hey everyone, welcome to the video lecture for Unit 4, Module 9.1. In this one, we're going to be talking about fluid compartments, fluid movement, and fluid electrolyte and acid-base balance. As always, please see the learning objectives here and utilize those to help you study for both the lecture quiz and the unit exam. All right, let's go ahead and get started with this one. So the next time you see a small pond, think about the fish in it. They live their entire lives totally dependent on the quality of the isolated environment. Severe water pollution will kill them, but even subtle changes can have equally grave effects. Changes in the volume of the pond, for example, can be quite important. If evaporation removes too much water, the fish become overcrowded. The oxygen in the food supplies will run out, and the fish will suffocate or starve. The ionic concentration of the water is also crucial. Most of the fish in the freshwater pond will die if the water becomes too salty. Fish in a saltwater pond will die if their environment becomes too dilute. The pH of the pond water, too, is a vital factor. This is another reason that acid rain is such a problem. Your cells live in a pond whose shores are the exposed surfaces of your skin. Most of your body weight is water. Water makes up about 99% of the volume of the fluid outside the cells, and it's also an essential ingredient of the cytoplasm. All of a cell's operations rely on water as a diffusion medium for the distribution of gases, nutrients, and wastes. If the water content of the body changes, cellular activities are jeopardized. For example, when the water content reaches very low levels, proteins denature, enzymes cease functioning, and cells die. In this module, we'll discuss the homeostatic mechanisms that regulate ionic concentrations, volume, and the pH in the fluid surrounding the cells. To survive, we must maintain a normal volume and a composition of both the extracellular fluid, or the ECF, which is the interstitial fluid, plasma, and other body fluids, and the intracellular fluid, or the ICF, which is the cytosol. The ionic concentrations and the pH, or the hydrogen ion concentration, of these fluids are as important as their absolute quantities. If the concentrations of calcium or potassium ions in the ECF become too high, cardiac arrhythmias develop and death can result. A pH outside the normal range can also lead to a variety of serious problems. Low pH acidity is especially dangerous because hydrogen ions break chemical bonds, change the shapes of complex molecules, disrupt plasma membranes, and impair tissue function. In this module, we're going to explore the dynamics of exchange among the various body fluids and between the body and external environment. Stabilizing the volumes, solute concentrations, and the pH of the ECF and the ICF involves three interrelated processes. The first is fluid balance. You're in fluid balance when the amount of water that you gain each day is equal to the amount of water that you lose to the environment. The maintenance of normal fluid balance involves regulating the content and distribution of body water in the ECF and the ICF. The digestive system is the main source of water gains. Metabolic activity generates a small amount of additional water. The urinary system is the primary route for water loss under normal conditions. Sweating can become important when body temperature is elevated. Although cells and tissues cannot transport water, they can transport ions and create concentration gradients that are then eliminated by osmosis. 2 is electrolyte balance. Electrolytes are the ions released when inorganic compounds dissociate. They are also named because they can conduct an electrical current in a solution. Each day, your body fluids gain electrolytes from the food and drink that you consume. Your body fluids also lose electrolytes in urine, sweat, and feces. For each ion, daily gains must balance daily losses. For example, if you lose 500 milligrams of sodium in urine and sweat, you need to gain 500 milligrams of sodium from food and drink to remain in sodium balance. If the gains and losses for every electrolyte are in balance, you're said to be in an electrolyte balance. Electrolyte balance primarily involves balancing the rates of absorption across the digestive tract with the rates of loss by the kidneys, although losses at sweat glands and other sites can play a secondary role. 3 is your acid-base balance. You're in acid-base balance when the production of hydrogen ions in your body is precisely offset by their loss. When acid-base balance exists, the pH of body fluids remains within normal limits. Preventing a decrease in pH is the primary problem because your body generates a variety of acids during normal metabolic operations. The kidneys play a major role by secreting hydrogen ions into the urine and generating buffers that enter the bloodstream. Such secretion takes place primarily in the distal segments of the distal convoluted tubule, or the DCT, and along the collecting system. The lungs also play a key role by eliminating carbon dioxide. Much of the information in this module should be familiar to you from discussions in earlier modules and from 168 about the aspects of fluid, electrolyte, or acid-base balance that affect specific systems. In this particular module, we'll provide an overview that integrates those discussions to highlight important functional patterns. This module has a wide-ranging clinical importance, so steps to restore normal fluid, electrolyte, and acid-base balances must be a part of the treatment of any serious illness affecting the nerves, cardiovascular, respiratory, urinary, or digestive systems. Because this module builds on information in earlier modules, we'll go back and highlight a lot of things that can provide a quick review. The image here represents an overview of the body makeup of a 70 kilogram or about 154 pound person with a minimum of body fat. The distribution is based on overall average values for males and females from ages 18 to 40. Water makes up about 60% of the total body weight of an adult male and about 50% of that of an adult female. This difference between the sexes reflects the proportionately larger mass of adipose tissue in adult females and the greater average muscle mass in adult males. Adipose tissue is only 10% water, whereas skeletal muscle is about 75% water. In both sexes, intracellular fluid contains a greater proportion of total body water than does extracellular fluid. Exchange between the ICF and the ECF takes place across plasma membranes by osmosis, diffusion, and carrier-mediated transport. The largest subdivisions of the ECF are the interstitial fluid of the peripheral tissues and the plasma of circulating blood. Minor components of the ECF include lymph, cerebral spinal fluid, synovial fluid, serous fluids, which are the pleural, pericardial, and peritoneal fluids, the aqueous humor, the perilymph, and the endolymph. More precise measurements of total body water provide additional information on differences between the sexes and distribution of body water. The greatest variation is in the ICF, as a result of the differences in the intracellular water content of fat versus muscle. Less striking differences occur in the ECF values due to variations in the interstitial fluid volume of various tissues and the larger blood volume in males versus females. In clinical situations, it's customary to estimate that 2/3 of the total body water is in the ICF and 1/3 is in the ECF. This ratio underestimates the real volume of the ECF, but that underestimation is appropriate because these proportions of the ECF, including the water in the bone, in many dense connective tissues and in many of the minor ECF components, are relatively isolated. Exchange between these fluid volumes and the rest of the ECF takes place more slowly than does exchange between the plasma and the other interstitial fluids. For this reason, they can be safely ignored in many cases. Clinical attention usually focuses on the rapid fluid and solute movements that accompany the administration of blood, plasma, or saline solutions to counteract the blood loss or dehydration. Exchange among the subdivisions of the ECF occurs primarily across the endothelial lining of capillaries. Fluids may also travel from the interstitial spaces to plasma through lymphatic vessels that drain into the venous system. The kinds and quantities of dissolved electrolytes, proteins, nutrients, and waste in the ECF vary regionally. Still, the variations among the compartments of the ECF seem minor compared with major differences between the ECF and the ICF. The ECF and the ICF are called fluid compartments because they commonly behave as separate sections. The presence of a plasma membrane and active transport at the membrane surface enable cells to maintain internal environments with a composition that differs from their surroundings. The principal ions in the ECF are sodium, chloride, and bicarbonate. The ICF contains an abundance of potassium, magnesium, and phosphate ions, plus large amounts of negatively charged proteins. This image compares the ICF with the two major subdivisions of the ECF, or the plasma and the interstitial fluid. If the plasma membrane were freely permeable, diffusion would continue until these ions were evenly distributed across the membrane. But it does not, because plasma membranes are selectively permeable. Ions can enter or leave the cell by only specific membrane channels. Also, carrier mechanisms move specific ions into or out of the cell. Despite the differences in the concentration of specific substances, the osmotic concentrations of the ICF and the ECF are identical. Osmosis eliminates minor differences in concentration almost at once because most plasma membranes are freely permeable to water. The only noteworthy exceptions are the apical surface of the epithelial cells along the ascending limb of the nephron loop, the distal convoluted tubule, and the collecting system. The regulation of fluid balance and electrolyte balance are tightly intertwined because the changes in solute concentrations lead to immediate changes in water distribution. Physiologists and clinicians pay particular attention to the ionic distributions across membranes and to the electrolyte compositions of body fluid. Four basic principles are key to understanding fluid balance and electrolyte balance. 1. All of the homeostatic mechanisms that monitor and adjust the composition of body fluids respond to changes in the ECF and not in the ICF. Receptors monitoring the composition of two key components of the ECF, the plasma and the cerebral spinal fluid, detect significant changes in their composition or volume and trigger appropriate neural and endocrine responses. This arrangement makes functional sense because a change in one ECF component will spread rapidly through the extracellular compartment and affect all of the body's cells. In contrast, the ICF is contained within trillions of individual cells that are physically and chemically isolated from one another by their plasma membranes. For this reason, changes in the ICF in one cell have no direct effect on the composition of the ICF in distant cells and tissues, unless those changes also affect the ECF. 2. No receptors directly monitor fluid or electrolyte balance. In other words, receptors can't detect how many liters of water or grams of sodium, chloride, or potassium the body contains, nor can they count how many liters or grams we gain or lose throughout the day. But receptors can monitor plasma volume and osmotic concentration. The plasma volume and osmotic concentration are good indicators of the state of fluid balance and electrolyte balance for the body as a whole, because fluid continuously circulates between interstitial fluid and plasma, and because exchanges occur between the ECF and the ICF. 3. Cells cannot move water molecules by active transport. All movement of water across plasma membranes and epithelia takes place passively. In response to osmotic gradients established by active transport of specific ions, such as sodium or chloride, you may find it useful to remember that water follows salt. As we saw in earlier modules, when sodium and chloride ions or other solutes are actively transported across a membrane or epithelium, water flows by osmosis. This basic principle accounts for water absorption across the digestive epithelium and for water conservation by the kidneys. 4. The body's content of water or electrolytes will increase if dietary gains exceed losses to the environment, and will decrease if losses exceed gains. This basic rule is important when you consider the mechanisms of fluid balance and electrolyte balance. Homeostatic adjustments affect the balance between urinary excretion and dietary absorption. Circulating hormones regulate renal function. These hormones can also produce complementary changes in behavior. For example, the combination of angiotensin II and aldosterone can give you a sensation of thirst, which stimulates you to drink fluids, and taste for heavily salty foods. These hormones mediate physiological adjustments to fluid balance and electrolyte balance. They are 1. Antidiuretic hormone, or ADH, 2. Aldosterone, and 3. The natriuretic peptides ANP and BNP. We introduced these and discussed these hormones in earlier modules. The hypothalamus contains special cells known as osmoreceptors, which monitor the osmotic concentrations of the ECF. These cells are sensitive to subtle changes. A 2% change in osmotic concentration, or approximately 6 milliosmoles, is enough to alter osmoreceptor activity. The population of osmoreceptors includes neurons that secrete ADH. These neurons are located in the anterior hypothalamus. Their axons release ADH near fenestrated capillaries in the posterior lobe of the pituitary gland. The rate of ADH releases varies directly with osmotic concentration. So the higher the osmotic concentration, the more ADH is released. Increased release of ADH has 2 important effects. 1, it stimulates water conservation by the kidneys, reducing urinary water losses and concentrating the urine. And 2, it stimulates the hypothalamic thirst centers, promoting the intake of fluid. The combination of decreased water loss and increased water gain gradually restores the normal plasma osmotic concentration. The secretion of aldosterone by the adrenal cortex plays a major role in determining the rate of sodium absorption and potassium loss along the distal convoluted tubule in the collecting system of the kidneys. The higher the plasma concentration of aldosterone, the more effectively the kidneys conserve sodium. Because water follows salt, the conservation of sodium also increases water retention. As sodium is absorbed, chloride ions also follow, and water follows by osmosis as sodium and chloride ions move out of the tubular fluid. Aldosterone also increases the sensitivity of salt receptors on the tongue. This effect may increase your consumption of salty foods. Aldosterone is secreted in response to increasing potassium or decreasing sodium levels in the blood reaching the adrenal cortex, or in response to the activation of the renin-angiotensin-aldosterone system. As we saw in earlier modules, renin release occurs in response to any of 3 changes. They are 1, a decrease in plasma volume or blood pressure at the juxtaglomerular complex of the nephron, 2, a decrease in the osmotic concentration of tubular fluid at the DCT, or as we'll see, 3, decreasing sodium or increasing potassium concentrations in the renal circulation. The natriuretic peptides, atrial natriuretic peptide and brain natriuretic peptide, are released by cardiac muscle cells in response to abnormal stretching of the heart walls. Increased blood pressure or increased blood volume causes this stretching. Among their other effects, these peptides reduce thirst and block the release of ADH and aldosterone that might otherwise lead to water and salt conservation. The resulting diuresis, or the fluid loss by the kidneys, decreases both blood pressure and plasma volume, eliminating the stretching. At first glance, it can be very difficult to distinguish between water balance and electrolyte balance. For example, when you lose body water, plasma volume decreases and electrolyte concentrations increase. Conversely, when you gain or lose excess electrolytes, you also gain or lose water due to osmosis. However, it's often useful to consider fluid balance and electrolyte balance separately because their regulatory mechanisms involved are quite different. This distinction is absolutely vital in a clinical setting, where problems with fluid balance and electrolyte balance must be identified and corrected promptly. Water circulates freely within the ECF compartment. At capillary beds throughout the body, hydrostatic pressure forces water out of the plasma and into the interstitial spaces. Some of that water is then reabsorbed along the distal portion of the capillary bed, and the rest enters lymphatic vessels for transport to the venous circulation. Fluid also moves continuously among the minor compartments of the ECF. So, 1. Water moves back and forth across the mesothelial surfaces that line the peritoneal, pleural, and pericardial cavities, and through the synovial membranes that line joint capsules. The flow rate is significant. For example, about 7 liters of peritoneal fluid are produced and reabsorbed each day. However, the actual volume present at any time in the peritoneal cavity is very small, less than 35 milliliters. Water also moves between blood and cerebrospinal fluid, between the aqueous humor and the vitreous humor of the eye, and between the perilymph and endolymph of the internal ear. The volumes involved in these water movements are very small, and the volume and composition of the fluids are closely regulated. For these reasons, we'll largely ignore them in the discussion that follows. Water movement can also take place between the ECF and the ICF, but under normal circumstances, the two are in osmotic equilibrium. For this reason, no large-scale circulation occurs between the two compartments. A small amount of water moves from the ICF to the ECF each day as a result of mitochondrial water generation, but we'll consider that topic in a separate section. We cannot easily determine the body's water content. However, the concentration of sodium, the most abundant ion in the ECF, provides useful clues to the state of water balance. When the body's water content increases, the sodium concentration of the ECF becomes abnormally low, and when the body's water content decreases, the sodium concentration becomes abnormally high. In our discussion of capillary dynamics, we considered the basic principles that determine fluid movement along the divisions of the ECF. Recall that the exchange between plasma and interstitial fluid, by far the largest components of the ECF, is due to the relationships between the net hydrostatic pressure, which tends to push water out of the plasma and into the interstitial fluid, and the net colloid osmotic pressure, which tends to draw water out of the interstitial fluid and into the plasma. It results in the continuous filtration of fluid from the capillaries into the interstitial fluid. This volume of fluid is then redistributed. After passing through the channels of the lymphatic system, the fluid returns to the venous system. At any moment, interstitial fluid and minor fluid compartments contain approximately 80% of the ECF volume, and plasma contains the other 20%. Any factor that affects the net hydrostatic pressure or the net colloid osmotic pressure will alter the distribution of fluid within the ECF. The movement of abnormal amounts of water from plasma into the interstitial fluid is called edema. Pulmonary edema, for example, can result from an increase in the blood pressure in a pulmonary capillaries. Generalized edema can result from a decrease in blood colloid osmotic pressure, as in advanced starvation, when plasma protein concentration decreased. Localized edema can result from damage to capillary walls, such as bruising, the constriction of regional venous circulation, or a blockage of the lymphatic drainage, such as in lymphedema. You lose about 2,500 milliliters of water each day through urine, feces, and insensible perspiration, or the gradual movement of water across the epithelia of the skin and lungs. The water loss is due to sensible perspiration, or the secretory activities of the sweat glands, vary with physical activity. Sensible perspiration can cause significant water deficits, with maximum perspiration rates reaching 4 liters per hour. Fever can also increase water losses. For each degree that a body temperature rises above normal, daily insensible water losses increase by 200 milliliters. The advice, drink plenty of fluids for anyone who's sick, has a definite physiological basis. Water gain is about 2,500 milliliters per day is required to balance your average water losses. This value amounts to about 40 milliliters per kilogram of body weight per day. You obtain water through eating, about 1,000 milliliters, drinking about 1,200 milliliters, and metabolic generation about 300 milliliters. Metabolic generation of water is the production of water within the cells, primarily as a result of oxidative phosphorylation in the mitochondria. When cells break down 1 gram of a lipid, 1.7 milliliters of water is generated. Breaking down proteins or carbohydrates yields much lower amounts, about 0.41 milliliters per gram and about 0.55 milliliters per gram, respectively. A typical diet in the United States contains 46% carbohydrates, 40% lipids, and 14% protein. Such a diet produces approximately 300 milliliters of water per day, about 12% of your average daily requirement. A rapid water movement between the ECF and the ICF in response to an osmotic gradient is called a fluid shift. Fluid shifts occur rapidly in response to changes in the osmotic concentration of the ECF and reach equilibrium within minutes to hours. If the osmotic concentration of the ECF increases, that fluid will become hypertonic with respect to the ICF. Water will then move from the cells into the ECF until osmotic equilibrium is restored. The osmotic concentration of the ECF will increase if you lose water but retain electrolytes. If the osmotic concentration of the ECF decreases, that fluid will become hypotonic with respect to the ICF. Water will then move from the ECF into the cells, and the ICF volume will increase. The osmotic concentration of the ECF will decrease if you gain water but do not gain electrolytes. So to summarize, if the osmotic concentration of the ECF changes, a fluid shift between the ICF and the ECF will tend to oppose that change. The ICF acts as a water reserve because the volume of the ICF is much greater than that of the ECF. In effect, instead of a large change in the osmotic concentration of the ECF, smaller changes occur in both the ECF and ICF. Dehydration and drinking a glass of tap water are two examples that demonstrate the dynamic exchange of water between the ECF and the ICF, as we'll now see. Dehydration, or water depletion, develops when water losses outpace water gains. When you lose water but retain electrolytes, the osmotic concentration of the ECF increases. Osmosis then moves water out of the ICF and into the ECF until the two solutions are again isotonic. At that point, both the ECF and the ICF are somewhat more concentrated than normal, and both volumes are lower than they were before the fluid loss. Because the ICF has about twice the functional volume of the ECF, the net change in the ICF is small. However, if the fluid imbalance continues unchecked, the water loss of the body water will produce severe thirst, dryness, and wrinkling of the skin. Eventually, a significant decrease in plasma volume and blood pressure occurs, and shock may develop. Conditions that cause severe water losses include excessive perspiration, as when you exercise in hot weather, inadequate water consumption, repeated vomiting, and diarrhea. These conditions promote water losses far in excess of electrolyte losses, so body fluids become increasingly concentrated, and sodium ion concentrations become abnormally high, a condition called hypernatremia. Homeostatic responses include physiologic mechanisms, ADH and renin secretion, and behavioral changes, increasing fluid intake, preferably as soon as possible. Clinical therapies for acute dehydration include administering hypotonic fluids by mouth or intravenous infusions. These procedures rapidly increase the ECF volume and promote the shift of water back into the ICF. When you drink a glass of tap water or when you're given hypotonic solutions intravenously, your body's water content increases without a corresponding increase in the concentration of electrolytes. As a result, the ECF increases in volume but becomes hypotonic with respect to the ICF. A fluid shift then occurs, and the volume of the ICF increases at the expense of the ECF. Once again, the larger volume of the ICF limits the amount of osmotic change. After the fluid shift, the ECF and the ICF have slightly larger volumes and slightly lower osmotic concentrations than they did originally. Normally, this situation will be promptly corrected. The decreased plasma osmotic concentration depresses the secretion of ADH, discouraging fluid intake and increasing water losses in urine. If the situation is not corrected, a variety of clinical problems will develop as water shifts into the intracellular fluid, distorting cells and changing the solute concentrations around enzymes and disrupting normal cell functions. This condition is called hyperhydration, or water excess. It can be caused by 1. Drinking a large volume of water or the infusion or injection into the bloodstream of a hypotonic solution. 2. An inability to eliminate excess water in urine due to chronic renal failure, heart failure, cirrhosis, or some other disorder. And 3. Endocrine disorders such as excessive ADH production. The most obvious sign of overhydration is abnormally low sodium ion concentrations, or hyponatremia. This reduction in sodium concentrations in the ECF leads to a fluid shift in the ICF. The first signs are the effects on the central nervous system function. The individual initially acts drunk. This condition, called water intoxication, might sound odd, but it's extremely dangerous. Untreated cases can rapidly progress from confusion to hallucinations, convulsions, coma, and then death. To demonstrate the seriousness of the condition, in 2007, a 28-year-old woman died of water intoxication after entering a water drinking contest. called Hold Your Wee for a Wee, sponsored by a Sacramento, California radio station. Treatment of severe overhydration generally involves administering diuretics and infusing a concentrated salt solution that promotes a fluid shift from the ICF to the ECF and returns sodium concentration to near normal levels.